Welcome to the station crew. Enjoy your stay.
In the previous episode, we unlocked the portable chem dispenser board, so finally we can start doing some more interesting chemical projects. And using this opportunity, I want to take this request from Schrödinger to create a cow. You previously spoke about its creation, cytology. So cytology is today's topic. Cytology is a science about creating organisms from cells, essentially cloning. That is a sub-department of science that is usually situated in the xenobiology. And the first thing we need to do is to hunt for cells, to first understand which organisms we can create from the cells that we have available. For that, I will need a couple of items. First, biopsy tool. It will let us take some cells from meat, from different animals, etc. And we also need petri dishes. I will create like 10 of them. These are the containers for the cells. Other than that, I want to get a plunger. We will need it when we will grow the cells into an organisms. I will leave it here for now. And I also think that I will use the body bag. It will let us store some of the organisms' bodies. I will also leave it here for now. So let me go through the station and scavenge for cells. Here in the biolab, we have this swab already available, and this is an item that we can use to get the cell lines from different objects. And I also have another petri dish here, so I will take this one as well with us. There are neglected ferns, so these are the first items that we can scrub for potential cells. We got some ophidic cells, aphid cells, and neuroamphibian cells. I think these are bees, frogs, etc. We'll take a look at it closer later. So when I get an example of cell, I will use the swab onto the petri dish, and it will store the cells inside the petri dish, clearing out the swab. Let me also check the trash. So here there is a bacteria-rich moldy bread. It's also a good source of cells. Let's swab this as well. It gave us some slime particles and murine cells, which I believe is the mouse cells. That's good too. Other than that, I can see that there is a bee. Let's scrub the corpse of the bee. And it will yield us the aphid cells. Right, I'll check this fridge here. There is a moly biopsy. That is a Quest biopsy. It has some bovine stem cells and globular ooze particles. We will leave it here for now. I will also scrap this neglected fern. And in the second one, we got some cells that we didn't see before. We will try those out later. Here in the AIS satellite, we have this old rotten gibbs. They are also a good source of cells. We got some bovine stem cells, which are the cow cells, some globular myocytes and globular ooze, which are for slimes. In the second old rotten gibbs part, we got similar cells. Please note that the regular rotting gibbs do not yield cells. We need only the old rotting ones, like this. Here we have some more slime, mouse, and blob particles. And the last one contains similar ones, so nothing really special, but let's move on. Here we have a space carp corpse, and we can take its biopsy using the biopsy tool, and then dump the cells into an empty petri dish that will give us the copernite cells, which will let us grow space carps. So we have another bacteria-rich multi-pieces slice. So let's swab it too. And it gave us some different cells. There are candid cells, which I believe is a corgi, and similar ones that we had. But this one, I think the blettoid anthropod cells are the cockroach cells. They're not really useful for us, but we could use it for genetics because we could merge our DNA with the DNA of the cockroaches later. And this moisture trap is also a source of cells. So I will swab it too. Getting some new cubic ooze particles, which is a cubic slime. Right, we got quite a lot of cells. There are 4 petri dishes here and a full box, and also this one that was here from the beginning. To analyze the cells, you can use this microscope. It is also craftable, and it can be made from some glass, plastic, and flashlight. What you do is you put the petri dish with cells inside, and then you can take a look at what's the petri dish containing. So this one we can see contains 2 types of cells, the bovine stem cells, which will create a cow. So this is a moly biopsy, I guess. It's from the quest that we read before, where there was a scientist that was trying to revive a cow to get her delicious milk. And also we can see that there is some other type of cells, the globular ooze particles. They also require some reagents to be created. They have the virus susceptibility, which will reduce the growth rate. So let us analyze what this scientist was trying to do. He was saying that there is a bottle for saying solution for moly. Here are the 2 bottles. One of them is a solution for moly, and there is an experimental solution. One contains peptides and another growth serum. All right. And it was also saying that there is a broth next to the growing vat. So let's find this broth and analyze what's inside this broth. Here it is. It has quite a lot of reagents. So let's analyze what will happen if we just put this broth into the vat. So the broth contains protein. It's a basic reagent that is needed for almost everything that you grow in the cytology. And then there are some other reagents here. Let's take a look at what is needed for the cow. So cow requires 3 reagents. That is a protein, nutriment, and a cellulose fibers. We have protein here, cellulose fibers, and nutriments. So these 3 are required for the cow. Then there is some slime jelly, organic slurry, Vertum 4th, and liquid Gibbs. These are not good for the cow, but they are the required reagents for the globular ooze particles. So if we use this broth straight away, both of the cells will start growing from this petri dish, and we don't really want that. To avoid the growth of one of this type of cells, we need to exclude one of the required reagents. So I will need to split this cultivation broth into different beakers and use them separately. And this experimental solution and solution for moly, they also contain peptides and growth serum. We see that the growth serum is required for the cow, and the solution moly has peptides that is not needed for the cow. It actually is a good reagent for the globular ooze that will increase the growth of the second types of cells. So this scientist mixed the bottles again, and that's good that we have the microscope to analyze this. So let us go and split this broth into 2 different beakers. I have separated the required reagents, so the cultivation broth beaker now contains protein, slime jelly, organic slurry, Vertum 4th, and some liquid Gibbs. That is also good for this second type of cells. And the other beaker contains just protein, nutriment, and cellulose fiber. That is good for the cow. And I will actually also use these supplementary reagents. So into the beaker for the cow, I will add the growth serum to let the cows grow faster. 5 units will be enough. And these peptides are good for the second type of cells, and we will also add them into this other cultivation broth. So let's take this biopsy. And grow those things. So here we have a growing vat and an input gate connected to it. The growing vat and input gate contain nothing right now. What we need to do is we need to place the biopsy with bovine stem cells and globular ooze particles into this growing vat. It says that I put some sample to the vat. And then we need to decide which one we want to grow first. So first, let's create the cow. The cow is this beaker with protein, nutrients, and cellulose fibers. I'll put everything into the input gate. And I will enable the resampler so that we can get multiple cows. And let's take a look at how the process of growing goes. So we see that the number of reagents is getting reduced in the growing vat. There are some bubbles coming. And after some time, it spawns a cow. So we have a nice, beautiful cow. And after some time, we can get some milk from its udder. Let's put this cow here for now. And I'll also place this glass window here so that it will not run away. Okay, so this growing vat ran out of reagents, and it was not enough for the second cow, unfortunately. But let me use plunger and try the second type of cells. So I'll clear everything and try to spawn this second type of organism. The cells are still there, but let me try to use it once again just to be safe. And I'm putting this cultivation broth contents inside the input gate. Let me actually leave this space in case if it will be some bad organism that we don't want to face. So the reagents are being consumed. The solution is bubbling, and we can see that the globular ooze particles are getting some growth progress. And the cow does not have any progress because there are no required reagents. So it gave us some kind of grapes, but they're aggressive, so let's kill this before they damage anything. So it was some kind of a slime. Yeah. Not really useful for anything, but I don't know. Maybe we can try to revive it later and it will be our pet. Right, we got our cow that will give us some delicious milk. We got our shooting grapes that were aggressive, but they're now dead, unfortunately. Let me check other cells that we have to figure out what else we can use. So this petri dish contains some slime particles. I will definitely want to look into the slimes later, but they contain some other particles, the globular ooze and the murine cells, which are the mouse particles. Let me check if we have any other petri dishes with slimes. Right. Another organism that I want to make is slime, and we have 4 petri dishes with different cells. So let's analyze which one is best. First, I'm going to take this one that have only 2 types of cells. We can see that there are slime particles and also some cells that will let us create mouse, but they both have only the protein is required reagents, which means that if we have a broth that contains only protein, they both cells will grow. We can only make some of the cells grow faster or slower if we suppress the cells or add supplementary reagents to them. But let me check if we have any slime particles next to the cells that require something other than protein. So this one contains also the slime particles and murine cell, but there are also Wilbur ooze, not really different from the one that we just seen. But this one is containing the slime particles next to other two cell types, which require a lot of other reagents, which means that if we add just the protein and use this petri dish, we will grow only the slime particles. And that is the perfect petri dish that I want to use to grow slimes. So to grow slimes, we need a source of protein. We used what we had to grow that cow and the ooze that we killed. And usually you can get the protein by grinding meat. And the meat, you can get it from some corpses like xenomorphs that died there. Or you can butcher a monkey from a monkey cube and get protein from the meat of the monkey. But there's also another more humane source of protein. And let me show you this other option. For that, I will need a portable chem dispenser. Let me actually build it. And while we're at it, let's also make a reagent grinder, because it's a very good tool to grind the meat in the bulk, and we can finally build it because it was unlocked with the biological technology that we researched in the previous episode. So this is the chem dispenser. It provides usually the basic reagents, but when you upgrade it with a femto servo, it will also provide some more advanced ones that are acetone, ammonia, ash, diethylamine, and also the stable plasma that we created previously. And I want to use this chemical dispenser to make a synthetic flash that we will later grind with this reagent grinder into the protein. It comes with a very nice reaction search interface. Here we can just look up what we need, and I want to have some cryoxidone. It shows here the chemical details, and I want to find the reaction to create a cryoxidone. It shows that I need some stable plasma, acetone, and unstable mutagen. It also highlights the reagents here in the chem dispenser, but the unstable mutagen is not available by default. Let me click on the reaction for the unstable mutagen, and let me create it on our own. So I will use 5 units of chlorine, phosphorus, and radium. Then click on the cryoxidone again. And since we already have acetone, stable plasma here, and unstable mutagen in the beaker, I will just click on these two. And we get our 45 units of cryoxidone. So making chemicals with a chem dispenser is much faster than the old way that we did by mixing the chemicals from the bottles. The second reagent that we will need is the blood, and I can donate some blood from this monkey. I can do this by attaching the IV drip, changing the direction from injecting to drain, maximizing the flow rate, and wait until the blood is taken from this monkey. It has 200 units of blood, so 100 is okay. But just in case, we can give the iron pills to the monkey so that it will recover the blood levels faster. Right, we have our blood, we have our cryoxidone, and now we can just mix it. So I will just dump the cryoxidone into the blood container. And we got quite a lot of synthetic flesh. Synthetic flesh is not good for cooking. It has only 1/3 of nutrient capacity of the normal meat that you get from a cow. But it is very good for getting protein. And by mixing those reagents, we got 97 units of protein. So we can use this to grow some slimes. Now we use this petri dish with the slime cells onto the growing bed. I will enable the resampler so that we will get multiple slimes and put the protein that we have into this input gate. You can see how the cubic ooze particles and ghettoidioid anthropod cells are not growing, while the slimes have growth progress. So it means that everything is working, and we only need to wait for the few slimes to pop out. So we got a lot of slimes from this protein, and it's still cooking. It's only like halfway through. So these are the slimes that this diary was talking about. They are the future of science, and the cell lines can be found on anything moldy and rotting, and these small blobs have crazy mutation potential when properly fed. But when they are not fed, they become aggressive. So I want to kill those and extract their cores and then revive them back into gray slimes to work on the xenobiology. So let me close the curtains here. Slimes are very weak to anything cold and related to water. So if you use this fire extinguisher with 50 units of water, it becomes a very good tool to kill the slimes. So even though the extinguisher ran out of water, I have put them under the shower and they died. Let me collect all the slimes and extract the cores from them. To extract a core from a dead slime, you could use a slime processor that can be built by flipping the switch on the food processor, or you could also use the serger tools. So taking surgical drapes, initiating the core removal, then use the scalpel to cut the slime, and then with a hemostat extract the slime core. This operation will give you this gray slime extract, which can be turned into a living slime when you inject a plasma in it, or it can turn into a monkey cube, even 3 of those, when you put some blood inside. So this is the third source of the monkey cubes that we were talking about in the previous episode. Unfortunately, we didn't get any galioform cells to make chicken, but we have this egg box that I found in the fridge in the kitchen, and I can show you how you can make chickens even if you didn't manage to find the cells. You can just get the eggs and start throwing them, and in one of the eggs you might find a chicken. We are out of eggs, and we got 2 chickens from those eggs. They will eventually grow into normal chicken that can lay eggs if we feed some wheat to them. So this way we got our first slimes that we'll use to kickstart the xenobiology. We got our cow to get 100% purity milk from, and we will get our chicken that will spawn some natural eggs that can be used for good pastry that will be also 100% purity. And with that, this request is complete.